Hi everyone, in this video we will discuss high dissolution drugs versus low solubility drugs, dissolution strategy differences. High dose and low solubility drugs behave very differently in dissolution. High dose products need large sink capacity. Low solubility drugs need enhanced media or stronger hydrodynamics. Each presents unique scientific and regulatory challenges. Understanding these differences prevents OOS results and ensures robust method development. That's the foundation of reliable dissolution testing. Not all high dose drugs are poorly soluble. For example, paracetamol dissolves easily, but many high-dose drugs fall into VCS class 2 or 4 and need discriminatory method. So remember, high dose does not always mean low solubility, but they often overlap. Failure mechanisms are different, so methods must be tailored. Media selection depends on dose solubility balance, and regulators expect strong scientific justification for each case. That's how robust dissolution methods are built. To ensure reliable dissolution, B must maintain sink condition, at least 3 times of drug solubility. For high-dose drugs, the API load is huge, so media can saturate quickly and may require longer volumes of surfactants. For low solubility drugs, solubility is poor across physiological pH, so dissolution is slow, even at low doses. Here, we use pH adjustment or surfactant. So, method design must match the dose solubility challenge to ensure robust results. FDA discourages excessive use of surfactants unless strongly justified. For high-dose drugs, even 900 ml may not be enough, so larger volumes or special media must be scientifically defended. For low-solubility drugs, surfactants use faces greater regulatory scrutiny. In both cases, media selection must ensure sink conditions, discrimination, and robustness. That's what regulators expect for reliable dissolution methods. When the medium becomes saturated, the dissolution rate drops, creating a false failure signal. This leads to plateau profiles that are not formulation related. High API load can cause agglomeration or thick gel layer formation. Large doses can also shift media pH, so buffering must match the drug's acid base nature. That's why achieving true sink condition is critical for meaningful dissolution data. When sink conditions are difficult, increasing volume is preferred over adding surfactants if justified. Use 1000 to 1500 ml, strengthen the buffer to control pH drift and surfactants only if necessary. Optimize agitation, typically 50 to 75 rpm, and consider USP apparatus 4 when continuous flow provides near infinite sink condition. This ensures more reliable dissolution data. For BCS class 2 and 4 drugs, solubility is poor across the pH range. Their hydrophobic surfaces resist batting, slowing dissolution, so batting agents or mild surfactants may be needed to mimic physiological solubilization. Even with good media, release can remain slow and variable. This causes high vessel to vessel variability. So, method must be highly sensitive and discriminatory. That's critical for robust method development. Always use lowest effective surfactant level to avoid masking formulation differences. Choose mild surfactants like SLS at 0.1 to 1%. Whenever possible, use biorelevant media FASSIF or FESSIF to mimic fed and fasted state. Adjust pH to match API's ionization profile. You can increase agitation within acceptable limit and for tighter control. Consider USP apparatus 4 for stable hydrodynamics. This ensures sensitive and meaningful dissolution testing. Using overly strong media can make all formulations look the same, hiding real differences. That's why discriminatory power studies are essential. Low solubility drugs are the most challenging category in modern development. They often need a combined strategy: surfactants, higher volumes, and optimized hydrodynamics. For pH dependent drugs, use multi-stage dissolution. But be careful. Media that are too strong create a non-discriminatory method. The goal is to reveal differences, not erase them. High dose tablets release large amount of fines that can settle at the basal bottom. This causes conning and poor mixing. So select RPM carefully, typically 50 to 75. Increase speed cautiously to prevent artifacts. Always verify basal centering and pedal alignment. If sticking or floating persist, evaluate USP apparatus 1, the basket. Proper hydrodynamics ensures reliable results. For regulatory submissions, documentation and justification are critical. For high dose drugs, use larger volumes and stronger buffers and justify them. For low solubility drugs, justify surfactants, pH adjustment, and biorelevant media. For high dose and low solubility, use a combination approach, strong but not overly aggressive. Clear rationale is what regulators expect. Discriminatory method expectations are: must detect formulation process changes, the test batch with different hardness, different granule size, wonder lubricant variation. Avoid media so strong that all profiles look identical. Dissolution must be physiologically relevant and still discriminating. For high dose drugs, control saturation and pH drift. For low solubility drugs, improve batting and solubilization. When they overlap, use hybrid approach. Optimize hydrodynamics, media and sampling based on real-time observations, and always provide strong regulatory justification. That's how robust dissolution methods are built. Thank you for watching this video. In next video, we will discuss dissolution for poorly soluble BCS class 2 and 4 drugs. Please subscribe us for getting update on upcoming industry application oriented videos, and stay tuned because in this 62 video series, we will cover dissolution method development, validation, troubleshooting, and many real industry examples. Please share your queries in comment section. See you in next video. Thanks.